Welcome to the Wednesday afternoon lecture series at the NIH. I'm Claire Waterman, I'm Director of the Cell and Developmental Biology Center at the Heart, Lung, and Blood Institute, and it gives me a great pleasure and is a real honor today to introduce our speaker, Mike Rosen. Mike was originally scheduled to give the walls last year. We had to cancel that because of the emerging pandemic, and in the hopes that he could come in person this year, we invited him a year later, but that didn't work out. So we are thrilled that he decided to join us on Zoom after a year of Zoom fatigue, and I'm very happy to introduce him today. He leads the Department of Biophysics at UT Southwestern Medical Center, where he is the Mar, Nell, and Andrew Bell Distinguished Chair. He's also been an HHMI investigator since 2005, and as of last year, is now a member of the National Academy of Sciences. I've known Mike for several years. I got to know him about 8 years ago, I would say, through our summers at the Marine Biological Lab in Woods Hole, where we both participated in summer research collaborations, and our labs were just down the hall from each other. I subsequently had one of my PhD students go on to do a postdoc with Mike. So you could sort of say in a way that we're now part of the same science family. We've both parented a kid together separately. But before I knew him personally, I knew of his beautiful biophysics and biochemistry work on the structure and regulation of actin cytoskeletal regulatory proteins. However, Mike's not going to talk to you today about the structure of actin regulators. He's going to talk about a much more recent discovery he serendipitously made, one that I consider to be one of the most important discoveries in cell biology in my scientific lifetime. And in the course of those studies of those actin regulatory proteins in vitro, Mike made the remarkable discovery that in solution, mixtures of multi-domain adapter proteins and their ligands undergo a liquid-liquid phase separation, forming what is now known as biomolecular condensates, and which are now known to concentrate proteins and nucleic acids, separating them from the surrounding liquid phase in the cytoplasm, promoting their activities, all without a surrounding membrane. So it's subsequently been shown by many in the field that this is a major principle of subcellular organization that's involved in diverse cellular processes from RNA metabolism to signal transduction. Mike's lab used a range of tools and techniques, including in vitro and cellular microscopy and biochemical reconstitution, to investigate the underlying biophysical principles driving this phenomenon and to show what forming these condensates actually matters to protein-regulated activities. He received his undergraduate degree in chemistry, summa, from University of Michigan. He went to the University of Cambridge, where he got a postgraduate certificate in natural sciences. He then went to Harvard, where he studied under Stuart Schreiber to get his PhD in organic chemistry in 1993. He went on to do a postdoc at University of Toronto with Tony Pawson and Louis Kay, and then started his independent career in '96 at Weill Cornell Medical College in New York City, where he was an assistant professor and had a dual appointment at Sloan Kettering. He then moved in 2002 to UT Southwestern, where he is now, and assumed his current position, which is the professor and the inaugural chair of the Department of Biophysics. In addition to his recent election to the National Academy, Mike honors include the Wiley Prize in 2020, the Emil Kaiser Award from the Protein Society in 2018, and a PCASE Award way back in 1997. The title of Mike's talk today is Droplets, Droplets Everywhere, Cell Organization by Liquid-Liquid Phase Separation. Mike, thank you so much for taking the time, in spite of your Zoom fatigue, to be with us here today. And we look forward to a great lecture.
Well, thank you very, very much, Claire, for that really overly kind introduction. As everyone in the audience will hear, I owe Claire a great debt for her sending me her student, Lindsay Case, to my lab, who, as you'll see, did just wonderful, wonderful work there. It's really been a wonderful morning for me here, seeing some old friends and meeting some new faces and just hearing great science after great science after great science. And so it's been a pleasure to spend virtually the time with you all. And it's great to have the opportunity to tell you about the work that we've been doing over about the last decade to understand how cells are organized through this process of liquid-liquid phase separation. And as Claire said, this is something that really over time has seemed to really develop into a very widespread organizational principle in cell biology. So just to very briefly introduce this area, my lab has been interested in these cellular structures that we call biomolecular condensates now for about a dozen years. And these are, as Claire mentioned, by definition, compartments in primarily eukaryotic cells, probably also some in prokaryotes, that concentrate various macromolecules, proteins, nucleic acids, probably small molecules as well, in the absence of an encapsulating membrane. These structures are really found in diverse areas of biology, everything from signal transduction to membranes, to RNA metabolism, to gene regulation in the nucleus. It's almost impossible to open up a journal nowadays and not find some new area of biology that is impacted by these biomolecular condensates. It's also becoming increasingly clear that many condensates are involved in important human diseases, most notably neurodegeneration, also cancer, certain skin diseases, are either appear to be caused by changes in condensates or at least associated, the basic biology of the diseases associated with condensates. A key feature of these structures is that although they are persistent over time, their components are very dynamic and are constantly exchanging in and out of the structures, but yet somehow they're able to maintain distinct compositions, distinct identities, and obviously then distinct functions. In general, condensates are viewed essentially as an organizational intermediate between macromolecules on the nanometer length scale and canonical membrane-bounded organelles, which are on more the micron length scale. Now, when we first got into this area, as I said, about a dozen years ago, we set out a series of questions that we were hoping to be able to answer. One of them is, what is the physical nature of these structures? How is it that they're able to be persistent and distinct, yet constantly exchanging molecules with the surroundings? What is their chemical nature? What kinds of molecules produce them and lead to their different compositions? How can they be regulated, both in terms of existence and dissolution, or formation and dissolution, but also how can their compositions be regulated? And of course, what I might argue is really the key question in all of this is, what are their unique biochemical and cellular functions? And how do those arise from these physical properties and chemical nature? So what I'm going to do this afternoon is spend a little bit of time going over some of the history of my lab here, and show you the work that my lab and other labs did to address these first three questions: physical nature, chemical nature, and regulation. And then I'm going to tell you in a middle vignette a couple of more recent stories, one of which involves Claire's student, Lindsay Case, on this key question, I think, of biochemical and cellular functions that arise through this process of condensation. And then finally, I'm going to tell you the last few minutes about a completely new part of the cell, a completely new biopolymer that we've gotten into, and that is chromatin. And some hypotheses that we have about how chromatin may be organized also through this process of liquid-liquid phase separation. So I'll start by going back again about a dozen years, as Claire mentioned. My lab studied for many years signaling pathways that control the actin cytoskeleton. And we were particularly interested in the kinds of pathways shown here, and that is proteins in the Wiskott-Aldrich syndrome protein family, or the WASP family, and their ability to bind to and activate this actin nucleating machine, the ARP23 complex, to produce new actin filaments de novo from actin monomers. Now, suffice to say that we became very interested in understanding the oligomerization of WASP family proteins. We published a paper in 2008 that suggested that oligomerization would be a mechanism to drive activity towards the ARP23 complex. And as part of that interest, we started studying and became interested in a well-known ligand to one family member, the NWASP protein, and that ligand is called NCK. And the reason we became interested in it is that all WAS proteins have large proline-rich regions, which in the case of this NWAS protein shown here, there are roughly 9 binding sites for SH3 domains, sarcomology 3 domains. And NCK was a known ligand of NWASP at the time that contained an SH2 domain and 3 SH3 domains. And so we reasoned that if you mix together NCK and NWASP, you might get some kind of large oligomer. And again, I'm interested in that. What was surprising is that beyond getting an oligomer, when we mix these two proteins above a particular concentration, they phase separated. We got formation of all of these little liquid droplets that would float around in solution, sometimes fusing. The figure 8 up here seems to be 2 fusing droplets. And moreover, the proteins became very highly concentrated, on the order of 100-fold concentrated within these liquid droplets compared to the surrounding medium. And when we saw this, we immediately began thinking about some of the work that people like Claire had done, cell biologists had done, that showed that WAS proteins, when they act in cells, very often do so through forming various clusters at membranes. So for example, the NWAS protein is involved in forming structures called invatopodia in cancer cells. And these invatopodia structures begin with the assembly of the NWAS protein into these micron scale foci on the plasma membrane, which become sites of actin assembly, here you can see on the left, which then produce the finger-like projection, which is the invadopodium. And so we got thinking, perhaps the concentration that we see here through phase separation could essentially be the physical property or process underlying formation of these foci. Now, we ultimately came to understand this process of phase separation through polymer chemistry. Polymer chemistry dating back to the 1930s and 1940s, sort of the dawn of the polymer era. Because way back then, people were asking the following question. If I have some binary interaction to make a complex, and they were studying in particular the reactions of acids and alcohols to make ester bonds, to make polyester for the war effort. But this was a very much an abstract framing of the problem. 2 elements that interact. If those 2 elements are strung together, each in multivalent fashion, how do they behave? And what was shown in beautiful experiment and equally beautiful analytical theory is that systems of this ilk, multivalent interactions, give rise to what are called sol-gel phase transitions. And that is a transition from small complexes at low fractional binding, or in the case of non-covalent systems, as I'm trying to indicate here, low concentrations. At low concentrations, these form small complexes that are soluble in solution. Beyond some critical threshold concentration, the systems become capable of assembling into huge macroscopic polymers that are often gelatinous in nature, hence the name sol-gel. Anybody who's ever polymerized an acrylamide gel has done a sol-gel phase transition. Moreover, what polymer chemists of the same era figured out is that as polymers get longer, they become less soluble. So if I have my little Eppendorf tube here with the solution containing monomers, meniscus at the top, as those monomers polymerize, make larger and larger oligomers, eventually they reach some critical length and crash out, forming a separate phase in solution. Often because polymers are flexible, that separate phase was a separate liquid phase. And importantly, these 2 processes, sol-gel transitions and liquid-liquid phase separation, are thermodynamically coupled in non-covalent systems, such that the polymerization from the multivalency can drive an insolubility that leads to phase separation. And so, again, ultimately what we came to understand is that multivalent interactions in the proteins that we were studying could drive polymerization. That in turn decreases the solubility of the molecules and leads to this property of macroscopic phase separation. Now, lucky for us in terms of timing, just a few months after we had those initial observations of our multivalency-driven phase separation, an initial paper of 2 came out from Tony Hyman's lab and his then postdoc Cliff Brangwyn, who's now running his own lab at Princeton, showing that in cells, some condensates appear to behave as phase-separated liquids. And so the first paper published in 2009 was on P granules in the C. elegans embryo. Second paper in 2011 was looking at the nucleolus in Xenopus oocytes. And in both cases, what they showed is that these structures, compartments in these cells, underwent cycles of fusion and fission. They showed fluid-like movements in response to shear stress. They rapidly exchanged molecules with the surroundings, so all characteristics of liquid-like behavior. And moreover, their assembly and disassembly properties were suggestive of a critical concentration, again, consistent with this being a phase transition-driven process. So when the first paper came out, we started looking at what are the molecules that are in these liquid-like compartments. And sure enough, whether one looks at things like RNA granules or one looks at membrane clusters, one sees many instances of multivalent macromolecules interacting with other multivalent macromolecules, either proteins with RNA or proteins with other proteins. And so we began to think that perhaps this idea of multivalency-driven phase separation could explain not only the clustering of actin regulatory proteins, signaling proteins, but perhaps these condensates really writ large, more generally, these structures like P granules and other known condensates in cells. And so that led us to our hypothesis, this idea of concomitant assembly and phase separation of multivalent molecules underlying fundamentally formation of condensates in cells. And this was the subject of our paper published in 2012. Now, since that time, this idea of multivalency-driven phase separation has really been demonstrated in a very, very large number of different kinds of molecules. We and others have shown a number of different instances of multi-domain proteins assembling and phase separating. We've looked principally at molecules involved in actin assembly, but another example comes from Minji Zhang's lab, looking at proteins that compose the postsynaptic density of neurons. Again, they see the same kind of behavior. There's a tremendous amount of work on so-called intrinsically disordered regions of proteins. And again, the concept is basically the same as in multi-domain proteins, in that there is an intrinsically disordered protein can be thought of as a series of weakly adhesive elements strung together on flexible chains. The adhesive elements here, though, are much smaller and weaker. It can be things like cation-pi interactions, charge-charge interactions. My colleague at UT Southwestern, Steve McKnight, has done a lot of nice work showing beta-strand-like interactions of disordered proteins. These all, again, lead to assembly and phase separation. RNA and DNA molecules with repetitive base pairing interactions can also drive phase separation. And finally, I think an interesting example that's distinct from these molecular examples, even something as large as synaptic vesicle, which is of course much, much bigger than any of the molecules here, if you coat it with weakly adhesive proteins, can phase separate to form a fluid whose basic unit is this very, very large vesicle, but nevertheless forms through this same physical process of liquid-liquid phase separation. And I'm going to come back to that idea of the scale independence of this process when I get to chromatin near the end of my talk. Now, this idea of multivalency driving phase separation very naturally leads to modes of regulation, because again, we know from polymer chemistry that higher valence, more sticky bits in these disordered chains, and higher affinity of the individual interactions that promote oligomerization. Higher valence or higher affinity will promote oligomerization, allows greater degrees of oligomerization at a given concentration, and thus will promote phase separation. And so this, we have shown this in a number of systems, but the one that I've been speaking about earlier, the NCK-NWASP system, NCK, as I said, has 3 SH3 domains. NWASP has roughly 9 binding sites. Those phase separate, but one has to go to relatively high concentrations to achieve phase separation. However, the SH2 domain of NCK binds very often to multiply tyrosine phosphorylated proteins. And in this case, there's a protein called nephrin that I'll say much more about in a little bit. that has 3 phosphotyrosines. And so when NCK is arrayed on nephrin, the effective valence of the SH3 domain jumps from 3 to 9. What that means is that the phase boundary shifts, and the same concentrations that didn't phase separate in the 2 protein system now do phase separate in the 3 protein system. And again, we and a lot of other labs have shown that there are various covalent modifications that can be introduced into these intrinsically disordered regions to either strengthen or weaken the adhesive interactions, and again, pushing around the phase separation boundary. And so I would say that in 2021, we understand at this point a fair bit about sort of the key physical principles that drive formation of biomolecular condensates and how those lead to regulation. But this question of function, particularly of biochemical function, remains something that I think is still a very, very active area of investigation. And essentially, what are the biochemical functions that uniquely arise from phase separation? Not from the molecules themselves, but from the phase separation itself. What does that contribute to activity? And so in the next vignette that I want to tell you about, I'm going to tell you 2 stories from the lab where we've specifically addressed this what I think is really important question of function. So the first one comes back to essentially my roots in actin regulatory signaling. And it comes to a pathway that controls actin assembly in the podocyte cells of the kidney. And it's a pathway that starts with a cell surface receptor called nephrin, which has a large extracellular domain that I'm not showing here, as well as an intracellular tail that is phosphorylated on 3 separate tyrosines. And those tyrosines then bind to and recruit NCK, which then binds to NWASP, and as I told you before, leads to local activation of the ARP2/3 complex to produce new actin filaments, again, that assist in forming the filtration barrier in podocyte cells of the kidney. Now, this was a pathway that my previous postdoctoral advisor, Tony Pawson, showed in a 2006 paper, can assemble actin filaments in discrete puncta on cells. And so he showed when nephrin are induced into puncta, you can see the image here on the left, the green dots are the nephrin puncta. Those puncta assemble these long actin filament tails, and those seem to be important in stabilizing cell-cell adhesions in podocytes. And so we thought that this would be a good system to understand how phase separation might contribute to actin assembly to function. And in fact, this is a system that we've now shown in a number of different ways will undergo liquid-liquid phase separation. As I mentioned to you before, if you mix all 3 of these proteins together, nephrin, NCK, and NWASP, they will phase separate in solution. Also, though, if we attach nephrin to a supported lipid bilayer, that is a bilayer that's attached to a glass surface, a flat glass surface, we see that nephrin alone will form a relatively uniform layer on that membrane. But if we add NCK and NWASP to it, what you see is the slide here, or the image here on the right, where the nephrin has assembled into these micron scale foci that have many of the same physical and chemical properties as these 3D droplets do over here on the left, but they are now constrained to the 2-dimensional surface produced by the membrane bilayer. And we recently have demonstrated that this same effect can occur in cells, where if we target a kinase to nephrin at the plasma membrane, it will assemble into these foci with NCK and NWASP. And so in both in 3 dimensions and in 2 dimensions, this system will readily phase separate. Now, what we showed back in 2014 is that if we create these nephrin clusters on a supported bilayer with NCK and NWASP, if we then add fluorescent actin and the ARP2/3 complex, what we see in the middle row here is that the actin assembles into these, in this case, bright red foci. And if you compare the nephrin foci with the actin foci, you see that they're basically coincident. You can see a little bit of that in the merge here at the bottom. And so it was very clear from these data that the principal sites of actin assembly are the nephron phase separated clusters. But the question then becomes, why does the actin preferentially assemble there? One possibility is, well, sort of duh, it has to. That's where the signaling molecules are concentrated. But another more interesting possibility is that perhaps there's something different about the chemistry within those condensate structures on the membrane that enhances or changes their activity toward actin assembly. And so to get at that, a new postdoc in my lab, Lindsay Case, coming from Claire's lab, set up a quantitative assay to measure the rates of actin assembly in this supported lipid bilayer system. And so again, what I'm showing here are the raw images. In this case, I'm imaging NWASP, but again, this is nephron NCK NWASP clusters. And we can image them over time as actin plus ARP2/3 complex results in formation of actin filaments on the foci. Now, of course, when we monitor this with a fluorescence microscope, we can, in one image, we can look at the rates of actin assembly inside the puncta versus the rates of actin assembly outside the puncta. And we can quantify those by looking at the maximum slope of these actin assembly curves. And what is immediately obvious is that the actin assembles, again, much faster inside the puncta compared to outside. But also, because we can simultaneously measure the local fluorescence of NWAS, we can correct the rate of actin assembly. We can divide the rate of actin assembly by the local density of NWAS. And what we see is that even when we normalize the polymerization rate to the amount of NWAS there, we still see appreciably higher actin assembly rates inside the puncta than outside the puncta. And I should note that most of the assembly outside the puncta is ARP2/3 independent. And so it's actually not just a 3-fold difference here in versus out, it's closer to a 14-fold difference in versus out. So the activity is much higher in the clusters, even when we normalize for NWAS. So what that means is that the specific activity of NWAS is higher inside the puncta. So that answers my first question. It's not merely that there's more NWAS there. Even when we account for the higher density, there's still an additional boost in activity. And so the question is, where does that come from? Now, we connected with Jay Groves, who's a biophysicist and single molecule spectroscopist and microscopist at UC Berkeley. And through interacting with him, came to understand that one of the things that's different between inside the clusters and outside the clusters is the amount of time that molecules remain on the membrane, so-called dwell time. And what we find is that for both NWASP and the ARP2/3 complex, the dwell time, the average dwell time, is appreciably higher inside the clusters than outside the clusters, even on the same membrane. And we can see that both by looking at fluorescence recovery after photobleaching on the left, or we can do single molecule imaging and actually count and quantify the lifetimes of individual molecules. And you can see again that inside the clusters, there's this long tail of long-lived molecules that you don't see if you look outside the clusters. And we came through additional experiments to understand that this higher dwell time results from the increased connectivity of the NWAS molecules in the ARP2/3 inside the condensates than out. There's just more bonds that need to be broken inside a condensate to release a molecule from the membrane than on the outside, outside the condensates. And moreover, if we then examined the relationship between dwell time and this specific activity, we saw this very, very, frankly, surprisingly good correlation between, again, the specific activity, the normalized polymerization rate, and the mean dwell time of NWASP, and we saw the same thing with ARP2/3 complex. And Lindsay was able to vary dwell time in a number of different ways in different experiments. And so what's shown here on the right is an overlay of a series of different experiments that perturb dwell time in different kinds of assays. And again, we see basically the same correlation that spans over these 3 different ways, these 3 different assay setups. And so essentially we came to understand this through the lens of kinetic proofreading. Without going into too much detail, suffice to say, we understood that the idea is that NWASP has to remain at the membrane long enough for the ARP2/3 complex to become activated to produce a new filament. So what I haven't told you is that the activation of the ARP2/3 complex is this long, many-second, multi-step process. And the idea is that if NWASP gets that process going, but then the whole system falls off the membrane before it gets to the end, it has to reset back to the beginning of the process. And in systems like that, kinetic proofreading theories tell us that the key parameter is the dissociation rate constant. And so again, dwell time here, the dissociation of the membrane, is the key controller of specific activity. And so our conclusions from this set of work are that phase separation increases membrane dwell time of signaling molecules, and that specific activity correlates with this dwell time, analogous to classical kinetic proofreading. And that therefore, as dwell time goes up because of condensation, activity goes up because of condensation. And I think it's important to note that these behaviors are likely to be very general. There are lots of signaling systems that are put together through modular interactions with multi-domain proteins that require multi-step processes to be able to achieve some signal at the end. And in fact, Jay Groves published back-to-back with us a study of signaling, of activation of RAS signaling, which shows essentially identical behaviors to what we've observed here. So let me change gears to a different functional system, and this is some work that literally was just published 2 days ago now. And in this case, we've looked at a completely different system, a different system of enzymes. And it's a system of enzymes that attaches SUMO, the small ubiquitin-like modifier protein, to a variety of substrates. And this is very much analogous to the way ubiquitin is attached to substrates. And the idea here is that there's a cascade of enzymes. There's an E1 enzyme that activates SUMO and that translates then that activated SUMO to the E2 enzyme. And the E2 enzyme in these cases transfers the SUMO directly to a substrate. And the question we asked is, if we recruit this enzymatic system into phase-separated droplets, what happens to activity? And so a student in my lab, Ben Peoples, was able to create synthetic phase-separated droplets with one of the model systems we worked in, a poly-SH3 protein that binds to a polyproline-rich motif protein. We mix those together, they phase separate, and if we rig those in a way, that they have what's called an FRB domain on them, an FKBP rapamycin binding domain on them, they will make droplets that can recruit E2 and substrate enzymes that are equipped with the FK506 binding protein, FKBP, on them, only when the small molecule rapamycin is added. So we can essentially make droplets here, and all of the enzyme system molecules remain uniformly distributed. We can add rapamycin to the system, and then the enzyme and E2 will get recruited into the droplets and be concentrated there. And when we do this, we were able to quantify the activity per unit volume inside the droplets and in the surrounding bulk. And what we found is that the droplets are much, much more active per unit volume than the bulk. When we quantify here, we see roughly a 36-fold increase in activity inside the droplets than in the surroundings. Now, again, this leads us to the same question that we asked with the Actin system. How much of this is due simply to increased concentration of these molecules in the droplets, and how much might be due to some other effects that act on top of this so-called mass action? And to do this, we essentially quantified the entire system. So in the droplet system, we use fluorescence microscopy to quantify the concentration of E1, E2, substrate, and also the SUMO protein inside the droplets. And then we could create those same concentrations of all of those components now in free solution in the absence of droplets. So basically we have a matched system here. We have two enzyme systems, both at the same concentration, but one of them, the molecules float around in free solution, and the other one, they are constrained within the droplets. And what we see is that even when we match the concentration, the rate of reaction is higher than in the droplets than in free solution, roughly 1.6-fold more active in droplets. And so again, what this tells us is that in addition to concentrating molecules, the droplets also enhance reaction rate by other mechanisms. And I don't have time to tell you the details, but suffice to say in this case, what happens in the droplets is that by tethering the E2 and the substrate to the scaffolding molecules that phase separate, we decrease the apparent Km of the reaction. And that then gives us this additional 1.6-fold boost in the droplets compared to those same concentrations in solution. So just to wrap this part of my talk up, I've told you about two different biochemical systems, one that controls actin assembly, one that controls assimilation activity. And in both cases, phase separation increases activity by concentrating molecules, kind of the intuitive increase you would expect, but also by additional effects that are specific to this mode of concentrating molecules. And the actin assembly system, that additional effect is the change in membrane dwell time. In the SUMO system, that added effect is a decrease in the apparent Km of the molecules. And so, we've gone 2 for 2. The two systems that we've studied both show these additional effects. My bet is, and this is more intuitive than anything else, is that this is going to be found to be true in other kinds of condensate systems. That condensates both take advantage of mass action, but then have other ways of modulating activity, likely tuned by evolution to further enhance or even suppress activity. So I've told you a little bit of history. I've told you a little bit about function. In my last few minutes here, I want to tell you about a new research direction in my lab to understand the potential role of phase separation in chromatin organization. And I'll spend just a moment kind of introducing the problem here, and then I'll tell you how we got into it in some of our data. So the fundamental problem in chromatin organization is that every cell of the human body has roughly 2 linear meters of DNA that has to be compacted down into a 10-micron diameter nucleus, so an incredible degree of compaction. But yet this compaction must occur in a way that retains the dynamics of the chromatin to permit regulation and function. And moreover, that compaction doesn't occur isotropically, it occurs anisotropically to create distinct functional domains on kilobase to megabase scales. And this organization controls essentially all nuclear processes, transcription, replication, repair. Clearly it goes awry in human diseases, particularly things like cancer. So clearly these mechanistic issues, how does all of this occur, are very, very important in the biological settings and human health settings. Now, if you look in textbooks, this organization has been explained through hierarchical models. And the idea there is that you start with a linear double-stranded DNA. The first order of compaction is that the DNA wraps around an octamer of histone proteins to form structures called nucleosomes. Then the model posits that those nucleosomes assemble into what's been called the 30-nanometer fiber. The 30-nanometer fiber then assembles into higher order fibers, which assemble into still higher order fibers, and ultimately you get something like the mitotic chromosome, the most compact unit of chromatin in eukaryotic cells. Now, despite sort of the intellectual satisfaction of this model and early data supporting it, as imaging has gotten better and better and better, and people have been able to look inside cells at the organization of chromatin, it's very clear that nucleosomes exist. They're easy to see nucleosomes in nuclei, but all of these higher order fiber structures have been quite difficult to observe in cells. And so I think it's the case that this question of higher order organization is still quite open in this field. So how did we get into this? Well, this comes back to this idea of scale independence of phase separation, because you can view chromatin as simply a long multivalent polymer. Each nucleosome is the basic unit, and they're strung together in a large array. The DNA wrapping around the histones is obviously very base or very acidic. All of the histone proteins have these long tails that emerge from the faces of the nucleosomes. Those are very basic. And so you can imagine basic acidic interactions running along the length of the polymer could give rise to phase separation. Moreover, we know that there are many different epigenetic marks, post-translational modifications that are put onto the histone tails. And many of the proteins that bind those, many of the reader proteins, are themselves multivalent. So one can think of multivalency in chromatin in that light as well through epigenetic modifications. And so even though this scale is very different from modular domain proteins, nucleosomes are gigantic compared to an SH3 domain. And even though DNA linkers are much, much stiffer than the polypeptide chain, the essential multivalency is the same. And so we've been thinking for many years, perhaps chromatin will phase separate. And this would be very much consistent with data in the literature for over 20 years now, showing that when chromatin is transferred into high concentrations of magnesium, it will phase separate, or sorry, it will precipitate. And in fact, when the few papers we've seen where people actually image this precipitate, it looks like it's sort of these globular kinds of structures. And so when a new postdoc came into my lab, Brian Gibson, who came from a background in genome organization, he took it upon himself to look at this issue. And so he was able to put together a dodecameric array of nucleosomes based on the Whittam 601 sequence, label them with fluorophores. And sure enough, when we transfer those into physiologic monovalent salt or also divalent salts, we saw formation of these beautiful spherical structures that contain both the histone proteins in the octamer as well as double-stranded DNA. These structures showed all of the hallmarks that we looked at of multivalency-driven phase separation. We could see a sharp switch as we increased salt concentrations from no droplets, the unmarked intersection points here, to droplets, all of the dark circles here. We could see sharp dependence on nucleosome concentration as well. If we varied the valence of the array from tetravalent to hexavalent to dodecavalent, 4, 6, 12, we saw that phase separation occurs at lower salt concentrations as we go up in valence. Interestingly, the degree of concentration that we see in this chromatin system is much, much higher than we see in other systems. We see about a 10,000-fold concentration of nucleosomes in the droplets compared to the surroundings. And what was satisfying to us is that the concentration of nucleosomes inside the droplets is sort of smack dab in the middle of estimates of nucleosome concentration in cells. Now, of course, I understand that doesn't mean that this is what's going on in cells, but at least we're not off by an order of magnitude. We're in the same ballpark. Now, these structures very clearly are dynamic liquids. We can photo bleach the center of them, and they recover on timescales of roughly 10 minutes. And so the fact that they recover tells us that they're viscous, that it takes about 10 minutes tells us that the liquid is highly viscous. But nevertheless, the nucleosomes remain quite dynamic within these structures. Now, one of the questions that we asked of ourselves early on is, are there instances in cells where people observe this kind of droplet-like behavior of chromatin? And the answer turns out to be yes. There are at least 3 that we're aware of. One of them is in apoptosis. So it's been observed for many, many years that when apoptosis is induced in cells, one sees condensation of the DNA as one of the early events. During keratinocyte development, or more generally, enucleation processes where the nucleus is ejected from the cell. Again, right before enucleation, one sees condensation of chromatin into these sort of spherical objects in the nucleus. And finally, if one looks at the macronucleus of tetrahymena, it's been known since the late 1970s that there are what are called chromatin bodies shown here in this EM image, which are sort of these spherical objects in the macronucleus. These are the sites of heterochromatin and tetrahymena. The surrounding lighter areas here are euchromatin. Now, what's interesting is that the one thing that unites all three of these processes is that when they occur, the DNA is being fragmented. That's what happens during apoptosis. And in fact, in this same paper on apoptosis, if you isolate nuclei and treat them with a variety of nucleases, you can see that in itself gives you condensation in the nucleus, condensation of chromatin. In keratinocyte, one of the processes that occurs is fragmentation. In the tetrahymena macronucleus, the heterochromatin or the chromatin is naturally in relatively small pieces. That's the way they package their genome. And so what we think this is telling us is essentially without long-range constraints, i.e. when DNA is chopped up, this liquid-like behavior manifests. And so what we think the relationship is between the droplets that we're observing and natural chromatin is that we're looking at short-range behaviors, interactions between relatively local elements of nucleosomes. Now, we've done a lot of work to understand how this phase separation behavior might be impacted by molecules or processes known to affect organization of chromatin in cells. We've looked at histone H1 binding, nucleosome spacing, and also histone tail acetylation and tail reader proteins. Now, in the interest of time, I'll just tell you in the last couple of minutes here about some work that we've done looking at this issue of acetylation. Now, as I suspect everyone here is aware, acetylation is a process that's known to open chromatin in cells. It essentially passivates chromatin towards processes like transcription and DNA repair. And so to understand how acetylation might affect our droplets, Brian rigged up essentially a system where we put a DNA sequence here, the TET operator sequence, into our nucleosome arrays. And we fused the catalytic domain of the P300 histone acetyltransferase protein to a transcription factor, TET-R, that binds with high affinity to that DNA recognition site. And as you can see in the images here on the right, the chromatin droplets are indicated by a fluorescent histone. We've attached GFP to our acetyltransferase, and you can see that transferase gets recruited quite well into the chromatin droplets. And so then the question is, what happens if we add acetyl-CoA, which is the cofactor of the enzyme? And you can see that as the reaction proceeds, the droplets remain liquid, but get dimmer and dimmer and dimmer, and then basically disappear and go away. And so what this tells us is that acetylation of the histone tails essentially destabilizes the interactions that promote phase separation, causing disruption of the structure. And we think that this perhaps is the biochemical or structural analog of the opening of chromatin observed in cells. Now, in just the last little bit here, last piece of data I want to show, we've now looked at this issue of histone tail readers, because acetylated tails are recognized by a particular domain in the cell called the bromodomain. And so we've looked at two different bromodomain proteins and their interaction with the acetylated chromatin. One is entirely synthetic. We made a pentabromo protein just by stringing together a single bromodomain. We've also looked at a natural bromo protein called BRD4, which has two bromodomains as well as self-assembly region. In both cases, we see the same thing. We can start with our chromatin droplets. We acetylate them with the histone acetyltransferase and they go away. But if we add a polybromo protein to them, those droplets reappear, or a new kind of droplets appears, which is this acetylated bromophase, both with the pentabromo protein at the top and also with the BRD4 protein at the bottom. Now, what was perhaps most exciting to us in all of this is if we start with a mixture of unacetylated chromatin and acetylated chromatin, the unacetylated chromatin phase separates, the acetylated one is uniform in solution. If we add the pentabromo domain protein to it, we see now formation of that acetyl-bromo phase. But if we overlay the two images, we see what we've taken to calling the chromatin caterpillar. We see that the two phases, the acetyl-bromo phase and the unacetylated chromatin phase, will adhere to each other, but they don't coalesce. And so what this tells us is that phase separation has produced two distinct chromatin domains with two distinct chemical compositions and two distinct physical properties that abut each other, but are different enough that they don't coalesce. And so this says that phase separation in principle could produce distinct chromatin domains in cells. Of course, again, I understand doesn't mean this is what happens in cells, but it does show that phase separation would be sufficient to doing this, and probably I would say then does contribute in some way to formation of distinct chromatin domains. Something you might imagine we're actively pursuing in the lab. So let me just wrap up by reiterating the conclusions from this last part of my talk. I've shown you that liquid-liquid phase separation is an intrinsic property of the chromatin polymer that can produce droplets that are highly compact, but yet retain dynamics that would be necessary for function. We've studied a number of regulatory factors. I've told you about acetylation and bromoproteins that provide mechanisms to create distinct chromatin domains with distinct chemical properties, distinct physical properties that could be used to functional effect in cells. So again, we're very interested, and this is a completely new direction in my lab, but we're interested in understanding a lot of the implications of this, as well as the physical and structural basis for these various properties. So let me just wrap up by thanking the people I've had the good fortune to work with on this project. All of the early work was done by 3 graduate students in the lab: Sudeep Banjare, Wei-Chin Cheng, Pilong Li. Lindsay Case was the postdoc, came from Claire Waterman's lab, who did the wonderful work on the function of condensation of actin assembly systems. Ben Peoples is a graduate student in the lab who did the work on the simulation system. And Brian Gibson is a postdoc, and Linda Doolittle, our lab manager working with Brian, did all of the work on the chromatin phase separation that I told you about. I mentioned Jay Groves has been a great collaborator and colleague on some of this work. I didn't have time to tell you about the work that we've done with Daniel Gerlich and his student Max Schneider and Cy Redding, who've done a lot of very interesting cellular work to explore this issue of phase separation of chromatin in sort of natural biological systems. So I thank all of these people for their wonderful work and collegiality on this project. Again, thank you, Claire, for the opportunity to spend the day at the NIH, and of course, all of you for your kind attention. Thank you.
Thank you, Mike. That was a great talk. Really cool. I will actually start the questions by asking you about the surface of these things. So I mean, that's the one thing that they have that's really different, right? Is there any evidence that there's something going on that when you're talking about the ones that are in 2 dimensions, they're bringing things in proximity to the membrane and increasing their dwell time there. Is there any thought that the interface between the condensate and the surrounding liquid is a special place that things might happen at where enzymatic activities get enhanced?
Yeah, so that's something that people are very, very interested in understanding. It becomes particularly relevant when you think about very small condensates. For example, those involved in transcription. Right? When you're looking at a giant, 5-micron diameter condensate, most of the material is in the center. When you're talking about a 200 nanometer condensate, the fraction that is in the surface is much, much, much larger. And so if the chemistry is different at the surface, yeah, you're going to see differences compared to if you look at something large. The truth is nobody's really looked at that experimentally, at least as far as I know yet. We have one interesting result that begins to speak to it. The chromatin droplets, when they encounter each other right after you make them, basically, boom, fuse very fast. If you wait 2 hours, what you see is that they stick to each other and they'll dance around and dance around for many seconds, and then they fuse. And what my physics colleagues tell me is what that means is that over time, the surface
tension's increasing. Yep.
has changed a lot.
Yep.
And so that says we're interested in understanding the structural basis for that, but that tells me that the surface is different from the rest. So yeah, I'll bet we see things there. We just got to find the experiments to do it.
Okay, now I will read some questions that have come into the from the audience, and what you were talking about actually is a nice lead into a question we got from two different people, which is about the size of these condensates and what limits the size on the supported lipid bilayers, but also in 3 dimensions. Is there anything known about that?
So there is. So we have seen on the supported lipid bilayers that if you have an actin cytoskeletal mesh on the membrane, you can see that the condensates will move around in the interstitial spaces in the mesh, but don't pass through the walls. And so basically that limits the ability of the droplets to coalesce in 2-dimensional systems. Cliff Brangwyn has shown the same thing in 3D in the nucleus. So when he disrupts nuclear actin in the Xenopus oocyte nucleus, which is very large, I think it's Cajal bodies basically will settle and coalesce into one large blob over time. Whereas if the cytoskeleton is there, they remain separate. So it seems as though active processes will limit the size of these things in cells in both 3 and 2 dimensions.
So if you go back to the idea that the size and the surface area have some possible effect on function, then you could see the crosstalk between the cytoskeleton and these.
Exactly.
Yeah.
Exactly.
Okay. Let's see. There is a question about whether, yes, does actin polymerization affect droplet morphology or other material properties as filaments grow inside or off the surface of condensates?
Yeah, also an interesting question. It depends on their composition. So I won't go into the signaling details, but with droplets composed of one set of signaling molecules, they do what I just described. They'll bounce off the walls. A different set of signaling molecules, including NCK actually and NWASP, those ones, if they encounter a wall, will wet the wall. They will wet the actin filaments. And so in that case, as the filaments form, and as the filaments get larger and larger, you'll see that the droplets themselves elongate along the filaments. And so there is relationship there, but it depends on the composition.
Let's see here. Okay, this was another one about the size, what limits the size. Phase separation is clearly dependent on the concentration. Apart from the local accumulation of protein, has anyone looked into whether phase separation events and how it coincides with increase in protein expression? So does protein expression level, can you induce phase separation by activating transcription and expression of proteins?
Certainly the answer is yes. Certainly in model systems, people have looked at, actually we've done it with some real systems, we haven't published it yet, the presence of condensates as a function of expression level over a population of cells. And in the few systems that people have really done this quantitatively, you see that below some expression level, you don't see visible condensates. And then there's a reasonably sharp threshold above that you do. And the size, the average size of the condensate grows as expression level increases beyond that. And so certainly one can think that over long time scales, expression level time scales, one can see regulation of condensates that way.
Okay, does the adhering of the acetylated and unacetylated domain happen due to some overall surface property acquired by the droplets, like an electrostatic charge, or what drives those guys sticking to each other?
Yeah, so the truth is I haven't had the hands in the lab yet to really explore that. My gut is that yes, as the questioner's intuition is that something about electrostatics and perhaps the distinct organization of the nucleosomes and the different kinds of droplets, but we really don't know at this point. It's something that we're hoping to get into.
Okay, I think we've reached the witching hour of 4:01 p.m. So I would like to thank you for really stimulating, cool talk and sharing the fresh off the press data with us. And it was a real pleasure having you, Mike.
Real pleasure to be here. Thanks again. Great to see you and great to see everybody that I spoke to.